Just continuing on with all these biomolecules and how the amino acids are used to make these biomolecules. And one of the first ones we're going to do is creatine, also related to phosphocreatine, which is really important. This is the wrong chapter. Which is really important for muscles in particular. First of all, it's a buffer system, and it's also used for energy. It's actually higher in energy than ATP. So you can actually take a creatine supplement. Of course, I always suggest that you don't take all these muscle supplements you don't want to take for long periods of time, because you'll just, in the end, downregulate your own endogenous production many times for creatine and carnitine and some of those. So that's why it's best to cycle on and off, which we are seeing something like it with cholesterol. Just like cholesterol does the feedback inhibition of its own biosynthesis, the same thing is true with a lot of these supplements in general. All right, phosphocreatine comes from creatine. And creatine comes from glycine and arginine. However, they may also say methionine, considering the fact that it's SAM, S-adenosylmethionine, also known as adomet. So that's why it's kind of, I don't want to say it's a trick question, but it requires those three things coming together. And so if I had asked you, what amino acids are used, and you said methionine, that is acceptable, because the adomet comes from methionine. And here it really isn't, I don't want to say tricky, this whole process. So we have glycine and arginine hooking up and kicking off ornithine. We've seen ornithine before. Where can that ornithine go?
To the citric acid? No.
Urea cycle. And the plus it's also used to make arginine, right? In a sense. And so this is another way that they interplay with each other. All right, this one has a very generic name. And once you see it, it makes sense because it has to do with the end of the arginine that's being added on to here. And in particular, here I'll just show it to you for time's sake. It's called an amino transferase. This is adding the end of that arginine transferring over to the glycine to make, if you remember your functional groups, it's called the guanidino group, the guanidino acetate group. Because we don't think it's a guanidino transferase, and the reason why it's not is for it to be a guanidino transferase, that nitrogen right there would have also had to be transferred. So it's only an amidino part that was transferred over to make a guanidino group that's on an acetate. This is where methionine, we've already talked about methionine being used, being adenosylated to make adomet or SAM. And the only difference between here and there is the fact that you're adding a methyl group. So the methyl group is coming from adomet to make adohomocysteine. Okay, and so what class or classes of enzymes can do this? Just in general, if you think about adomet or SAM, what class of enzymes are they involved in? Like, what's the whole purpose? Transferase, and what are you transferring? Methyl group. That's why I see it's just a methyl transferase. Now we make creatine, and creatine has its own functionalities. But the creatine and phosphocreatine is really important for energy. We have ATP used to phosphorylate the creatine. So what class or classes of enzymes could this be? It's a kinase. And if you were going to name this kinase, what could you name it? And it's creatine kinase. So that's why this one isn't out of left field, so to speak. The hardest one is trying to remember that it's an amidino group, because it's not really just an amide, it's an amidino group, so it's not in. That's why I've seen some people say like amino transferase or amide transferase, but it's amidino transferase, that first one. I actually haven't updated the next slide. I'm just giving a little disclaimer. I haven't updated the next slide this semester. And just this semester, just as of 2016 and 2017, there's been even more things discovered about it. And I don't think it's in your book. Some of the stuff that they're discovering is really, really recent. So, it didn't even dawn on me to update your the PowerPoints. But it has to do with glutathione. So those who have taken chemistry of cancer know we talked a lot about glutathione recently. And some of that stuff that they're finding out about glutathione and glutathione's role in carcinogenesis is really some of the reviews that are just coming out in the last year or two. And so that's why it's really recent. And I actually don't have it on here. But it is also important for carcinogenesis and the life cycle of the cell. Okay. But what is glutathione? I mean, just in general. First of all, is it big or is it small? It's relatively small in the big scheme. That's kind of relative, right? You know, it's not a Smurf, but it's not a human. You know, it's a poodle size. I don't know. Because what is it made up of? Do you remember? We had talked about glutathione briefly last semester. I don't know if we talked about it. There's a cysteine in there. I mean, it's 3 amino acids, okay? It's a tripeptide. And so it's a modified tripeptide. It has cysteine, glycine, and glutamate. Okay, in it. Right. And glutathione is really important because it's all about that cysteine. That cysteine can form disulfides to make that cysteine bond, that disulfide bond between the two. So it's got a lot of roles. I mean, it does lots and lots of things. And as I mentioned, in recent years of discovering that it's actually even more important than what they thought it was, because it's actually relatively in high concentration in your body, in some tissues more than others because of the fact of what it does. Okay. We've already talked about disulfide. Actually, I just mentioned sulfurs. You have to realize, remember, sulfurs, thiols in particular, are both good leaving groups and good nucleophiles, right? And so that's why they can form disulfides, whereas oxygen really doesn't. Oxygen can't. What happens when an oxygen forms an oxygen-oxygen single bond, which is what a disulfide is, right? There's 2 sulfurs coming together to form a single bond. What happens, what's it called when 2 oxygens come together to form a single bond? It's a peroxide, which forms ROS as a reactive oxygen species. And so, you know, that's not happening. Disulfides are fine, okay? But sulfurs themselves are also good nucleophiles, so that's why it's really important to keep the thiols that are supposed to be thiols. I said that right in my head. I don't know if I said it right out loud. But it's important to keep things that are supposed to have free thiols free, and things that are supposed to be disulfides. And you don't want them to accidentally interact, okay? Because you can imagine as a protein is first being made, there are lots of cysteines there, and you don't want the wrong cysteines to come together to form a disulfide bond. And glutathione helps with the folding in that sense. Glutathione is also a buffer, okay? Because sulfurs can be either reduced or oxidized, so it's an important buffer. It's also important for iron, to keep iron since it can gain and lose electrons. It's important for keeping iron in the oxidation state. And then one thing, like I said, that I did not add in, and we could actually have an entire class on, is that there's a metabolic pathway that's involved in, that's important in carcinogenesis. Or you can think of it, which is just, carcinogenesis just means cell proliferation that's gone out of whack. Okay, and what happens is glutathione, you have to have enough glutathione, a certain amount of glutathione around, and whenever that gets depleted, the cell should die. Okay, and so it's one of the new ways. Does anyone from chemistry of cancer know what's that new way of cell death? We say new; it's been around forever. But you know, what is it called? Ferroptosis, because it has to do with this whole interplay between glutathione and iron. And what happens is, whenever it goes out of whack, you can have an increase in reactive oxygen species because your whole iron metabolism's off. And it's different. It's separate from apoptosis and necrosis. Okay, it has its own. And right now, like I said, right now they're just trying to figure it out. The review that we've been going over is 2017, I think, that just came out this year, and there's some of the papers that are from 2015, 2016, but it's relatively new. It's a new form of programmed cell death. And whenever some things go wrong, then it's another way of carcinogenesis. And you can also think of it, this is also one way sometimes that cancer cells themselves cannot live forever, and they run out of glutathione, and when they run out of glutathione, it's their way of going through death as well. And then we're going to talk about how it's synthesized. Oh, whoops. This is 2 steps. It's nothing outlandish. There's just one important note, and for those in chemistry of cancer, we've actually already covered these since it's part of that metabolic pathway that's important for ferroptosis. This would be a healthy cell. Okay. It comes from glutamate, and glutamate's going to hook up with cysteine at the expense of an ATP. Okay. So, what class or classes of enzymes could this be? It could be a kinase if we're doing phosphorylation. What's else could be? Synthetase. And if you were going to name it, what would you name it? Good job. And now, one thing is you have to think back to last semester to remember whenever they named dipeptides, and also a carryover from organic chemistry, the first amino acid of the dipeptide gets that YL because it's a functional grouping of. So it's gamma glutamyl cysteine synthetase. If you said glutamyl cysteine synthetase and give you partial kind of all the credit. Does anyone know why is it important that it says gamma? What does glutamate have that only one other amino acid has? What does glutamate have? It's got 2 carboxylic acid groups, and that lets you know which one is forming the new peptide bond. So if you look down, I know it's going to give you it away, like the end product down there, you can see where the glutamate and the cysteine come together is not that normal peptide bond, where the backbone of the glutamate is connecting to the backbone of the cysteine. It's on its side. So it almost, to me, kind of looks, if you drew the hydrogen out, it kind of looks like a cross that's on its side, instead of 2 crosses being together, like what you normally see, where these are the R groups. But, yep. And so that's why they say gamma. It's like, normally this is the alpha amino acid, the alpha one. Well, I mean, that's the C alpha, but the carboxylic acids hang off the C alpha, and now it's hanging off of the gamma plate position. Okay, but then we have glycine that's coming in, and once again, we have the expense of an ATP to make glutathione. And so if you're going to name this, first of all, what class of enzymes can do this? Synthetase. If you're going to name it, it would be, and then that's exactly what it is. Oops. It's a glutathione synthetase. 2-step process. Relatively simple, nothing way out there, and like I said, this glutathione can go on to have a whole host of things. And for those interested, you can even look up, and I'll try to remember to incorporate that in. They're actually putting out a new textbook, and who knows, maybe the new textbook will go over ferroptosis in greater detail for the future, since it's a new pathway, relatively new pathway. Okay. Now we're going to one of my favorite, and I always really like to talk a lot about this. That's your neurotransmitters, because they are important. And it really ties chapter 22 to chapter 23. Ties them in together really, really well, since 23 is on the hormones. Chapter 23 is a little different than all the other chapters. Because in and of itself, it's no longer about a metabolic pathway per se, in like synthesis or the breakdown, but how do they interplay? Okay. Like if we get the chance, it goes over the actual role and how insulin works and gets released, like the physiological aspect of it. And if we have enough time, we also go to the physiological aspect of like leptin and ghrelin and cortisol and how they all interplay. I can't stress this the same. Just like I stressed in organic chemistry, how when you learn the carboxylic acid reactions, whether you're doing amides or esters or acyl chlorides, remember you always made it essentially intermediate, you collapsed it. It's the same thing over and over again. These is the key step is the same for all of these. that we're going to be talking about. So that's why once you kind of get an idea, you're going to see the same kind of thing over and over and over again. All right. So the first up, we're going to be talking about dopamine, norepinephrine, and epinephrine, which, what was another name for epinephrine? Is adrenaline. What's another name for norepinephrine? Noradrenaline. Okay. All right. So these are derived from tyrosine. That's why it's very important you make sure you get tyrosine in your diet. Just to do a little, as you get cut up, a little background. Of course, dopamine is really important with respect to neuromuscular disorders, things like dystonias, which I used to suffer from, Parkinson's disease. These are usually used in underproduction, whereas if you have too much, it's believed to be linked to some of the psychiatric and psychological disorders such as schizophrenia. Okay, it's a neurotransmitter. The other thing to pay attention is this is tetrahydrobiopterin. Okay, tetrahydrobiopterin is one that I don't think we've seen before, but it is a cofactor that's involved in redox reactions. And in particular, as we can see, you can see it with the redox reactions with respect to oxygen. Don't confuse it with tetrahydrofolate. That's the one, they kind of sound a lot alike, and I've seen people get mixed up. Tetrahydrofolate is the one that's for single carbon transfer and comes from folic acid. Tetrahydrobiopterin is for redox reactions. Okay, so this first step, we have tyrosine making dopa. Dopa is the precursor for dopamine. What are the only differences between tyrosine and dopa? It has an extra hydroxyl group. So if we think back, what class or classes of enzymes can do this? If you're adding a hydroxyl group, what could be a possible class of enzyme for that one? Don't overthink it. Okay, hydrolase. Hydrolase usually has to do with water, and there is a water involved. What's another possibility for hydroxyl group? If it was a carboxyl group, what would we call it? Carboxylase. So if it's a hydroxyl group, that's one of the possible names. And it's a hydroxylase. This one's a hydroxylase, and in particular, what specific hydroxylase would this one be? It's tyrosine hydroxylase. Please note it also technically is a monooxygenase. It's, I think, just one of the oxygen atoms to it. All right. Now we have dopa, and the only difference between dopa and dopamine, first of all, it uses our good friend PLP, which if you think about it, PLP, there are 3 types of reactions that PLP did. What was one of those reactions that PLP does? Aminotransferase. Remember, it's a cofactor in every single aminotransferase. What was the second type? Decarboxylation, which is what it's doing here. And the third type, remember, which is the one that we actually don't talk about a whole lot about, was racemic, switching between L and D amino acids. It is a racemate, which is called a racemase. Okay. So we're removing a carboxylate group in the form of carbon dioxide here, using PLP, and notice it's in the little, whatever color that is. What class or classes of enzymes could this be? A what? A carboxylase or decarboxylase. And this one's called it is a decarboxylase. So what could you name this one? I could see, dopa decarboxylase. What if I told you that it was promiscuous? Remember, we've had some of those promiscuous ones where they just kind of given a generic name. It is a decarboxylase, so but that's the last name of it. What does dopamine and dopa have in common? A phenol group, which what kind of compound is a phenol? Aromatic. This is an aromatic amino acid decarboxylase. It is very, promiscuous. It gets around. That helps you rebirth. This dopamine. Little side note, dopamine doesn't cross the blood-brain barrier. Where people who have certain dystonias and Parkinson's disease, they have to have an increase in dopamine. Does anyone know how do they do that? What's one of the drugs that they can take? What are the precursors? Dopa. Okay, and the problem with dopa is it is chiral, as you can see here from the first carbon. And so there's L and there's R and there's levodopa and carbidopa. One of them, levodopa, is the enantiospecific, and carbidopa, I believe, is the one that's the racemic mixture. But you have to take, it crosses, but not well. So whenever you first get sick with one of these diseases, they give you large quantities of dopa. And you take these horse pills, it seems like, to try to get enough dopamine across your brain to see if that helps. Okay. All right, so now we're doing norepinephrine and epinephrine. Of course, these are your fight or flights, and we've already talked about, I'm not going to actually talk about them because we talk about epinephrine a lot, adrenaline and noradrenaline a lot. Okay, we've got a new cofactor, ascorbate. What is it more commonly called? What? Well, the ATE in English means is a carboxylic acid that loses hydrogen. So when it would be a carboxylic acid, it's called ascorbic acid. But what's it more commonly called? I take a supplement of this every day. What vitamin is it? No. No, that's retinoic acid. Vitamin C! Come on, what have you talking? You gotta know your vitamins. At least some of them. Vitamin C is ascorbic acid. Vitamin A is retinoic acid and the retinol things. Vitamin K is vitamin K. That one, I'm sure it stands for something. And this K is for coagulation, it's German. But then vitamin D has a really big long name because it's the steroid one. Okay, and D and D3, so I don't. No, this is vitamin C. All right, but we can see here, once again, what's the only difference between dopamine and norepinephrine? Another hydroxyl group. So what do you suppose, what class of enzyme do you suppose this is going to be? It's a hydroxylase, and if you're going to name this one, dopamine hydroxylase. If you want to be even more specific and give the locant, the Greek locant for it, which carbon is being hydroxylated? I mean, if you told me dopamine hydroxylase, I'd give you points. So this one, you know how we have like alpha, beta, delta, gamma, all those? This is beta, right? Because the alpha is the one that's touching the nitrogen. This is the beta. And so if it's dopamine, beta hydroxylase. Probably from, if I remember right, the next one is the most difficult of the entire night, or entire day. We have from norepinephrine, or noradrenaline, to epinephrine, adrenaline. It uses adomet, which is just the same thing as saying S-adenosylmethionine or SAM to make I always want to say histamine, adohomocysteine, or adoHCY. What's the only difference between norepinephrine and epinephrine? The methyl group on the what? On the nitrogen. So what class or classes of enzymes can do this? This is an N-methyltransferase. So the N that gives you the locant. It is a methyltransferase. It's the name. And I mean, I can explain the reason why, but I don't like it. Okay. Phenylethanolamine N-methyltransferase. Why they don't say norepinephrine N-methyltransferase, I do not know. Okay. But you can see why this is called, this really is ethanolamine. And this is a phenyl group. I mean, my hypothesis is the fact that that's what the important part for that enzyme, and it probably recognizes more than just norepinephrine, but I can't say it for sure because I've never worked with it. But it's an N-methyltransferase, and all that N, once again, means, and you can tell it's italicized, is that's where the methyl groups, because in theory you have lots of places where you could stick a methyl group, but it sticks in that specific spot. So we made epinephrine. Life is good. We can run from the bear. Remember, what direction do you run from the bear? Downhill. Hey, if you've learned one thing from biochemistry, you learn how to run from bears. Just like in forensic science, I always say, don't look down the barrel of a gun. I say that so many times that people don't. All right, next we're going to move on to GABA, which is short. Some people don't know this. It's short for gamma aminobutyrate, also known as gamma aminobutanoic acid, or butanoate. It depends on if you use the IUPAC nomenclature versus not the common one. It's an inhibitory neurotransmitter. And remember those GABA A and GABA B receptors. I can't remember if we go over that in more detail next chapter or not. All right. So if we look at it, what compound is the starting material for GABA? Someone said it. Say it. What? It's glutamate. That's just plain Jane glutamate. Okay. Once again, we have PLP. The enzyme uses PLP. It's removing carbon dioxide. So what class or classes of enzymes can this one be? Decarboxylase. And if you're going to name this one specifically, it would be called? And it's glutamate, yes. Glutamate decarboxylase. We know it cannot be the aromatic amino acid because that's not an aromatic amino acid. Okay. Right, now we're going to, like the bane of my existence, histamine, and probably the bane of many of your existence, is a vasodilator. Of course, that's one that a lot of people have an allergic response to, hay fever, all that kind of stuff. What material is its precursor? As histidine, of course, we're removing we're removing a carbon dioxide using PLP. So what class or classes of enzymes can this one be? It's a decarboxylase, and if you were going to name this one in particular, it would be histidine decarboxylase. Once again, see, that's why I could say, and they even show you pink. That's the same step over and over and over. The only difference a lot of times between this amino acid and its neurotransmitter is that it's been decarboxylated. And then they may modify it from then, but that's the common theme. We just have a couple minutes, so I want to quickly go over serotonin in like 2 minutes. Okay. Just because we start here with the you can't see it, but what compound is the one that starts? Kind of got cut off. That's tryptophan. Good friend tryptophan, And tryptophan, okay, we see tetrahydrobiopterin being used once again. What are we making? So the only difference between it and they call it 5-hydroxytryptophan, which by the way they many times abbreviate as 5-HT. What class of enzymes could this one be? It's the same kind of principle that we've seen already a couple times today. It's a hydroxylase, and so if you're going to name it, it would be called, and it is, it's tryptophan hydroxylase. Okay, and then the last step is we have 5-HT. We're removing the carboxylate group in the form of carbon dioxide using PLP as a cofactor to make serotonin. So what class or classes of enzymes could this be? It's a decarboxylase. What one would this one be? Okay. What other one could it be? What class of compounds is tryptophan? It's aromatic. And so what one is this one? it's the one that gets around. It's the aromatic amino acid decarboxylase. And so you just have to remember, I mean, if you called it 5-HT decarboxylase, I'd give you most, if not all, the credit. It's just that that enzyme does accept a lot of the aromatic amino acids that can be decarboxylated. That's serotonin on your own. You can go through, we used to talk about it in maybe next chapter. We can go into better details with it. But that concludes